Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about regulation of gene expression in prokaryotes. In the previous video lecture we talked about the regulation of gene expression and we also talked the level of regulation of gene expression as well. But in this video we are going to talk about regulation of gene expression in prokaryotes. Now the regulation of gene expression in case of the prokaryote is a very simple. Inshallah, in the next video we will explain the regulation of gene expression in eukaryote, which is very complicated. In regulation of the gene expression in case of the prokaryote, there is, let's suppose we have a DNA. Inside a DNA we have genes. So different types of the genes we have. So these genes are actually controlled under one promoter. Promoter is actually the sequences or the region where the polymerase enzyme bind and it can, start the transcription and translation. The promoter, we can also say that the promoter is actually the region which can control the genes. There are multiple genes which are controlled by one particular promoter. Okay, so inside a DNA we have promoter and we have also the gene as well. It means genes X, Y, Z. These are, let's suppose, the genes. And when we get the RNA, and from RNA we get the protein. Okay, so this is very simple in case of the prokaryotes. After that, this protein are used for different purposes. Okay, so this is a simple process in case of regulation of gene expression in case of the prokaryotes. Let me a little bit explain it that what happened. Let's suppose we have media. Inside the media, what we have the lactose, which is actually a carbon source and which is actually used by the cell of the bacterial cell, let's suppose. So bacterial cell is actually need a glucose, not lactose. So what happened? The bacterial cell need a particular type of the enzyme or protein which can cause the breakdown of the lactose, and once the lactose are breakdown, so then we get the glucose and galactose. Now this glucose are actually need by the cell, bacterial cell. Okay, so for that purpose what happened, the bacterial cell genes will turn on and it will produce lac enzymes or lac proteins, which is lac Y, lac Z, lac A. Okay, these are the protein which are specially used for the breakdown of the lactose. So if, let's suppose, the lactose are present, so then the gene will turn on and it will produce lac proteins. When lac protein are produced, so then it will cause the breakdown of the lactose and we get the glucose, and the glucose are used by the cell, which is actually the energy source or the carbon source for the cell as well. One thing more, if there is a glucose present in a media, so the cell doesn't need lac proteins. If lac proteins are continuously produced, the cell doesn't need lac protein. So what happens? This glucose will bind on the promoter region of the bacterial DNA and it will cause the stop. It means it can stop the process of the protein, means that it can stop the process of the central dogma of the cell. Thus, and this way what happened, the protein will not form and the cell will not use that type of the protein. Now how the whole process will occur, let me explain it through a diagram. What happened, let's suppose we have a DNA. Let's suppose this is a DNA. Inside a DNA there is a specific portion. Let's suppose this is a specific portion which we call that promoter. Okay, and there are specific region. Let's suppose this is another region which we call that operator. Okay, and there are specific region which is actually different types of the genes. Let's suppose these are the genes. This is gene X, gene Y, or gene Z. Let's suppose these are the genes. So what happened, there are certain type of the protein which can bind on the promoter region, not exactly at the promoter region, at the enhancer region. There are some sequences which we call it enhancer sequences. At that particular type, the protein are activator protein. At that portion, the activator protein are bound, and it will cause to bring the RNA polymerase. RNA polymerase will bind, and it will cause the transcription process. Then what happened? We get the transcription, means mRNA. Let's suppose this is mRNA. Inside the mRNA, there are particularly sequences which is present for the synthesis of the proteins. So this process is actually called as transcription. Now, RNA polymerase bind here, and it will cause the means transcription of all these genes. As you can see, X, Y, Z, all these genes are transcribed. Okay, and then from that mRNA we get what? We get the protein, as you can see. This is, let's suppose, the protein. This is actually the protein X or Y or Z, or all these three protein we get. Okay, so this is actually the process. Here I want to tell you that how this process is actually working. This process is actually working by this promoter region. The promoter region is actually control these genes, expression of the genes. There is protein bind on the promoter region, so then the gene expression will occur. There is some protein which we call an inhibitor, which bind on this promoter region, which can cause the means the protein synthesis will be stopped. Okay, so this promoter region is actually control the expression of the gene. Operator region is actually that region which can, operate, or it actually give the signal to the genes to get ready for the transcription. This is the operator region. Okay, so this is actually the regulation of the gene expression in the prokaryote. So in the prokaryote, the gene expression is very easy. There is another thing which is very important. We call that operon. Operon. Actually, the operon concept, we will explain it in detail, inshallah, in upcoming videos lecture. We will explain operon concept in detail. Now, operon, in case of operon, what we have two type of the genes which we call it structural genes and non-structural genes. Non-structural genes. So this is actually the non-structural gene, and this is actually the structural gene. These two things is actually called as what? These two things is actually called as operon. So in case of the prokaryotes, the operon is very simple, but in case of eukaryote, the operon concept is very difficult, or is not difficult, means complicated. It's very complicated. So what happened? Now let me explain. Let's suppose we have a media. Let's suppose this is what this is. Let's suppose a medium. Inside this medium, we have what? We have a lot of lactose present. Lactose. A lot of lactose is present. Okay? Lac means lactose. A lot of lactose is present. What actually going on? The bacterial cell using glucose as a carbon source, or glucose for the energy, or glucose, it can use the glucose for the different purposes. For the energy purposes, the bacteria use what? Bacteria use glucose. Okay? Bacteria need what? It need glucose. Now inside the lactose we have what? We have the glucose. But how the bacteria doesn't need the lactose, but inside the lactose we have the glucose. Now how this lactose will be break down and it will produce the glucose. When the glucose are produced, so then the bacteria use it. Okay, so inside the media we have this all lactose. So what happened? Inside the bacteria, the DNA are present, as you can see. This is the DNA of the bacterial cell. So we have a promoter region. This lactose protein will lactose means carbon source are bind to this portion, to the promoter region of the cell, promoter region of the DNA. So when it is bind to the promoter region, so what happen? It will cause the genes to turn on, means regulation of the gene expression will be turned on. At that point, RNA polymerase, let's suppose this black is, let's suppose this is RNA polymerase. RNA polymerase will bind here, and it will, what it can do, it can produce mRNA, and from mRNA then we get the protein. This protein is actually, let's suppose, lac proteins. Inshallah we will explain lac operon. We have different types of the operon. We have lac, we have trap, we have rax, we have lax. Different types of the operon we have. Inshallah we will explain all these operon in detail. So what happened, this is, let's suppose, the trap, or this is, let's suppose, the lac operon. So in the lac operon, we get the lac protein. When we get the lac protein, this lac protein or lac enzyme will cause the destruction of the lactose. And when this is, let's suppose, the lactose, it will cause the destruction of this lactose, and from this lactose then we get glucose plus galactose. So when we get glucose and galactose, so what happened? The cell will use it. This bacteria cell will use it. So this is how the gene will be turned on in case of the bacteria, in case of the prokaryotic cell. If let's suppose, again, if let's suppose we have a media, let's suppose again, and in this media we have glucose present. Let's suppose glucose we have present. The cell using it continuously, but the gene is turned on, and what actually this operon this lac operon can doing, this lac operon can producing lac protein, which the cell doesn't need. Why? Because inside the cell, inside the media, glucose is present. So if the glucose is present, so the bacteria cell use it. So the lac protein is not important, because lac protein is important to break down the lactose to produce glucose, and then the cell use it. If inside the medium glucose is already present, so this lac protein is actually creating a problem, or this lac protein is actually extra. So what happened? The cell will turn off that process. So at this point, the glucose will come and bind at that particular area. So the glucose will bind here, and what it can do, it can turn off. So when the gene are turned off, so then we didn't get RNA and we didn't get the protein, or the protein we didn't get the lac protein. So if we didn't get the lac protein, so what happened? Lactose will be not break down. Although the lactose is not present, glucose is present, but in this case the process will be stopped. It means protein synthesis will be stopped, and the bacterial cell can easily use the glucose very easily. Okay, so this is how the regulation of gene expression occur in case of the prokaryotic cell. So that's all about the regulation of gene expression in case of the prokaryote. I hope this video helpful. If you like this video, make sure to hit the like button, share this video, and also subscribe the channel. And stay tuned, because in the next video we are going to talk about regulation of gene expression in case of the eukaryotic cell. Thank you so much for watching that video.